Hello everyone, we have come up with another experiment as part of our Do at Home experiment series. In this video, we are going to explore more about DNA. So if you are wondering what is DNA, DNA is basically deoxyribonucleic acid. It's a molecule that contains the instructions an organism needs to develop, live and reproduce. These instructions are found inside every cell. They are passed down from parents to their children. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, guanine, cytosine, and thymine. The order of these bases is what determines DNA's instructions or genetic code on how to build cells. To know the DNA sequence, we have to first extract the DNA. This can be done in laboratory or even at your home. So in this video, we are going to extract DNA from banana using the items that are available at your home. To conduct this experiment, we need bowl, rubbing alcohol, dishwashing liquid, salt, banana, water, rod or a spoon, strainer, and a glass. To start with, add banana into a bowl. Also add half cup of water and mash it well. You can even use a blender for this. After that, add half teaspoon of salt and 1 tablespoon of dishwashing liquid into the bowl. Mix it thoroughly. Use a strainer to separate the solution. In the next step, pour the distilled alcohol gently from the side of the glass. If you watch closely, you can see a fine layer at the top and a precipitation at the junction between two layers. That precipitation is DNA. Let's take a closer look into this experiment. Most wild bananas are diploid, just like humans. That is, they have two copies of each gene-bearing chromosomes, one from each parent. The soap molecule have hydrophilic part which loves water, so they are mainly attracted to polar molecules, and an hydrophobic part which hates water, and they are attracted to non-polar molecules. The cell membrane is partially polar and non-polar, and it is a phospholipid bilayer. When we mash banana, the cells in it get lysed. The hydrophobic tail of detergent get attached to the lysed cell, which has a lipid moiety, and pulls it into the water, just like how detergent removes oil and dirt from your clothes. The solution is left with DNA, which is later aggregated by salt. When we gently pour the distilled alcohol into the solution, the DNA is precipitated out in the alcohol layer. Here, the alcohol is less denser than water, so they are seen at the top. Since DNA is not soluble in alcohol, they are seen as precipitate at the junction between two layers. You can collect this precipitate with a rod or a spoon and observe it. That's it for this experiment. We hope you enjoyed this video. Thank you.